All right, but first I wanted to give a couple of impressions of Ann Arbor that I've had in the last day. This is my first time being here and I'm delighted to be here. And I'm really excited that this was such a beautiful campus to be on, all right? It's absolutely beautiful. And all right, great people. I've enjoyed meeting with so many people today and it's been absolutely delightful. A couple of additional observations, all right? One, your weather here is wacko. It was 85 degrees today and somehow or another, all right, this is what it was when I took this screenshot a little bit earlier today. Bonkers. I don't know what's going on. Secondly, your squirrels here are really scary. They're extremely large and very yellow with their tails. I don't get it. Something is wrong. And finally, it has been brought to my attention that Biostat does not have a designated elevator. I'm going to have a talk with Bramar about this. So Peter, expect a call. All right, so that being aside, all right, let's talk a little bit about work, and in particular, the human microbiome. Right? So over the last few years now, there's been tremendous interest in understanding the relationship between the human microbiome and health and disease. So by the human microbiome, we're talking about all of the bacteria that live in and on each and every one of us. All right, we all have microbiomes, and all right, you have one in your gut, you have one on your skin, you have one in your hair, you have one on your left pinky toe. All right, and the goal here is to understand how the composition of this microbiome is related to human health and disease. And there have been tons and tons and tons and tons and more tons of papers showing the relationship between the microbiome and disease, and also exposures. So exposures and things that influence microbiome include things like diet and exercise. You have drugs, smoking, alcohol, as well as broadly speaking, things like treatments, whether or not you receive medication and certain things, all right, all influence your microbiome. That's to say the bacteria that live in and on you. Right? Typically, we focus on a particular microbiome, such as the gut, all right, which is talking about the poo, or say the vaginal or the cutaneous skin microbiome. Then there's also been tons of studies showing the relationship between outcomes and the microbiome. All right, and so I put in a question mark here because in many cases, it's actually not clear whether it's the condition driving the microbiome or the other way around. All right, and that's something that simply you cannot discern from the cross-sectional studies that most people are conducting over here. It's a limitation of the data, but nonetheless, we oftentimes treat it as an outcome. And so this includes everything from asthma, cancer, diabetes, to even things such as treatment efficacy and patient survival. All right, we've found all sorts of associations with the microbiome. All right. And so how many of these studies worked? Classically, what you did is you got a bunch of healthy mice, all right? Then what you did is you got a bunch of unhealthy mice, all right? And so maybe you fed these unhealthy mice or these other mice their own body mass in NutraSweet, all right? That's actually a study, an example of something that people have done. And then from there, you collect their poo. And so from the healthy mice, you get happy poo. From the unhealthy mice, you get unhappy poo. And you know it's unhappy because it's frowning at you. And when my poo frowns at me, I know that there's something seriously wrong. Right? And then you compare their compositions. Very, very straightforward. And so with these traditional experiments, they tend to ask very, very simple questions. I've got my cases, I've got my controls. All right, what are the bacteria that are different between these two groups? Design, very, very simple. I have a bunch of mice in cages, and I basically compare the composition of the pellets that they are generating over here. Oftentimes, the number of individuals or organisms was very, very small. All right, and because of that, we were looking for astronomical effect sizes, which when you feed in any organism its own body mass in anything, you end up seeing very, very large effects. And so those are robust, those are identified, and people are very, very happy with those types of studies. The problem, however, is as we move to humans, all right, mice and humans are not the same thing. All right, and in fact, if mice were the same thing, we would have cured cancer about 50 times over by now. But the reality is what works in mice does not always work in people, and they are not the same organisms, regardless of what these bozos may think. So humans are way more complicated. In particular, when we conduct human studies, there's considerably more heterogeneity, with one major reason being that you're not allowed to lock people up in cages and feed them their own body mass in anything. All right. So I tried to figure this out. I said, hey, can we take graduate students and do this? And unfortunately, my IRB said, no. All right. I tried asking about postdocs, but they said that they go to animal care use. All right. But so I didn't really ask. I already knew that they were animals. All right, but the result of this, of course, is that many individual findings replicate very, very poorly. All right, people have found that, say, one bacteria is closely related to obesity, and then the next study says, no, 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 it's not. So many of these replicate poorly. People are not quite sure what to do. And a lot of this is driven by the fact that once you start talking about humans, all right, and a lot of day-to-day behaviors, your effect sizes tend to be very modest in size. And so in order to actually have any power to do anything, all right, we need much larger sample sizes. And so this has really motivated the development of large-scale microbiome profiling studies, where now we're collecting data on hundreds or thousands or tens of thousands of individuals. And so with this come a lot of opportunities. Fundamentally, it allows us to robustly and powerfully identify associations from our data. All right, your sample size is larger, you can start really digging into there a lot more carefully. Secondly, you can ask more sophisticated questions of the data. You can start talking about things like effect modification, mediation, and other strange and wonderful things that we make up in statistics. Right? Finally, a lot of these studies are conducted within the context of existing cohorts and large epi studies. And so there tends to be very rich metadata. So things like exposures, risk factors, outcomes, and so on and so forth. Oftentimes, we also have other omics, and there's a lot of interest in studying, say, the genetic basis for the microbiome and MBQTLs over here. And so some examples of large-scale studies that are out there include the CARDIA study, MS600, which at the time was quite large. The PIN study, 1,200, Hispanic Community Health study has 3,000, next 6,000. I was very proud of that. Then I gave the talk at NCI, and they were like, yeah, we've got more than 10,000. And then you go to Harvard, and they're like, yeah, we got 25,000. So. But they're Harvard and nobody likes them anyway. Okay, so from there, however, there's a lot of opportunities, but it also brings about tremendous challenges as well. So some of these challenges are inherent to the large sample sizes. So we're getting now hundreds, thousands of samples, but along with that, we get challenges that just because we're dealing with hundreds or thousands of samples. And so one example of this is batch effects. All right, and then there's also challenges that arise from the new opportunities. So now with these large sample sizes, we can do much more careful modeling. We can more robustly identify associations. And so how to do that now is a more significant challenge than when you only had 3 rats here and 3 rats there. All right, a t-test is probably as good as anything else you'll ever be able to do with that sample size of data. All right. So I'm going to talk about those 2 problems. And so the first part of my presentation is going to be about conquering batch effects with a method that we call conquer. I polled an audience once on whether it should be concur or conquer, but they said concur was too related to this travel thing, so they didn't like it. All right. So batch effects, what are batch effects? Batch effects are systematic variation in data that arise from differential handling or processing of the data in some way or another. So if you have a bunch of samples that were sequenced on one instrument versus another instrument, even on the same instrument over time, all right, or if some samples have been stored for longer than others, or even if a different technician processes your samples. All of these can result in serious batch effects, but it is systematic variation that results from differential handling or processing somehow. Right? And so the interesting thing about batch effects is that they can lead to false positives if it's not balanced, and they can also lead to false negatives by introducing additional variation into your data. And of course, these are not unique to microbiome data. They were first discovered within the context of proteomics, killed a couple of careers when things were subject to batch effects. Very unfortunate situation. But since then, we've recognized their existence and we deal with them using one of 2 strategies. One is both of them are called batch corrections, but one of them is about batch removal. And in batch removal, the idea is that you have batch and you try and remove the variability that is related to batch. And with that, you now have a clean data set for which you can do all of the strange and wonderful things you may be interested in doing. Batch adjustment is slightly different. It typically focuses on association analysis, and you're trying to adjust for having the data that were collected in batches. So both of them are called batch corrections, but there are slight differences between the 2. I'm going to focus entirely on batch removal in this case. All right, where I want to get a clean data set, so to speak. So here's an example of a data set that had serious batch effects. So it comes from the CARDIA microbiome study. So CARDIA is a very, very long time running study on risk factors of cardiovascular disease. And in their year 30 follow-up, they collected microbiome data on about 600 individuals. And they also have tons of covariates related to CVD, such as blood pressure, demographics, so on and so forth. And so this is what the data look like over here. So these are principal coordinate analysis plots. In statistics, we call them multidimensional scaling plots, which is basically like a PCA, except we use a non-Euclidean distance over here. And so what happens over here is each of these dots is a different sample, all right? And each color corresponds to a different batch over here. So a different sequencing batch. So the data will run across 6 different sequencing batches, and you can see that there's tremendous differences between the batches. The number is at the centroid of where the batch was based on the first 2 principal coordinates. This is using a dissimilarity matrix, dissimilarity called Bray-Curtis, and this one is using Euclidean on a transformed data scale. But regardless, overall, you see that there is considerable heterogeneity in the data due to different batch effects over here. And so how do we deal with batch effects? Well, they've been around for a long time. And so can we just borrow approaches from other omics data analysis, particularly DNA microarray analysis, right? So by far the most commonly used approach for batch correction is something that is called COMBAT, developed by Lou and my friend, Evan Johnson. It was actually a 2-month summer project that he did and is now one of the most highly cited papers in biostatistics. The idea behind it is very, very simplistic. It's simple. What they do is they take each gene, in their case, and then basically regress it on covariates and an indicator for batch. And then they basically remove the batch variable over here and get a corrected variable. It's a little bit more complicated, but that's the basic idea, especially if your sample size is large over here, that they're just regressing out the effect of batch, all right? So there are some generalizations for count data, but in general, these make very, very strong distributional assumptions. And then with microbiome data, when we're looking at taxa, which is the unit of analysis, what we typically have are count data. So these Gaussian approaches don't really make sense. And these generalizations for count data also don't really make sense, simply because they tend not to work for very ugly, zero-inflated, over-dispersed count data that are what microbiome data are over here. So there are a few approaches that have been tailored for microbiome data, but these oftentimes require very, very specific types of controls or spike-ins in order to do some types of correction. And overall, most of them are about batch adjustment rather than batch removal. And so the focus is almost entirely on association analysis rather than on getting a clean data set out there. So instead, we have our proposed method concept. And so here's the punchline of this part of the talk. These are the CARDIA data before. So this is the same slide, the same figure as what you saw before. And so this is before correction. And this is what happens after we do the correction. And so you can see that actually all of the data are smushed into the same place. And actually their overall distributions look very similar as well. So over here, you can already see the shape of each of these different batches was pretty similar. And that is maintained over here. All right, so before, after, okay. All right, with that, let's talk about the actual method itself. So CONQUER stands for conditional quantile regression. The idea is very, very straightforward. What we do is we correct each taxon separately. Specifically, we estimate the distribution of the taxon given covariates and batch, and then we simply remove the batch variable from there. So very, very much like COMBAT and these other approaches. Operationally, we use quantile regression in order not to avoid parametric, a lot of parametric assumptions here. And the idea is borrowed from COMBAT-seq, which is similar, except they assume a negative binomial model. By using quantile approaches in a zero-inflated 2-part model, it addresses the zero inflation and also ensures robustness to complex distributions and also allows for correction of higher order effects that are not simply means and variances in some cases. Right? So here's the 2-part model that we work from. So this is simply logit the probability that it is positive, so non-zero. And so it depends on some covariates as well as the effect of batch. And then this is the corresponding conditional quantile function over here. So it again depends on some covariates and the effect of batch over here. And so here, Y is the outcomes, W, we have to jigger because it's count data. And then here, we're just regressing it on the batch effects. And so the idea is that we're going to estimate these guys, and then we simply remove them from the model. Very, very straightforward, but very, very simple in concept. Operationally, for each taxon, what we do is we fit the two-part quantile regression model. There's a little bit of statistical messiness in the fitting, which I'm not going to go into great detail about because it is a zero-inflated model here. But it's still not terribly difficult. And from this, we can estimate the original quantile function of each sample for a given taxon. All right, so each sample now has its own quantile function over here. Then what we do is we estimate the corrected quantile function by simply removing the estimated batch effect. So nothing terribly complicated going on. You simply drop, you say, well, I'm going to assume that batch is equal to 0, and it falls out of your model over here. Then this is what we call the regression step. And then in a matching step, what we do is we locate the original observation and call that the tau hat quantile of the original observation. Then we pick the corresponding corrected quantile, the value of the corrected quantile function at the same particular quantile, and we set this as the corrected value. So, and we repeat this for all taxa. So this is done on a taxon by taxon basis. Operationally, what happens here, suppose this is the original quantile function over here, so the distribution function, and then this is the corrected quantile function over here. So this one has batch, and this is what happens after we remove the batch variable. All right, you can see that it's 0 and then becomes positive, same thing over here. So suppose the original value was 1500, then we come over here and you say 1500. This is up at the 80th percentile, 80th quantile, and then we come to the corrected one and we match it to the 80th quantile, come back over here. This is about 700. And so the corrected value now goes from 1500 to say 700. Right? So it's a simple quantile matching approach in this case, but it's done on a per sample, per taxon basis. Right? So let's give some of the estimation things. It's not really that complicated, but basically, we just have to deal with some discontinuity between the zeros and the first positive value, and we just do some linear interpolation there. So nothing terribly complicated. But this is again the overall thing. So we use a two-part model formulation. The logistic part deals with zero inflation. The quantile part deals with the non-zero components. And then from there, we also input additional characteristics and covariates. And then from there, we can get a conditional distribution estimate for the original distribution function and also for the batch corrected. And then we do matching in order to get the corrected values over here. So here are just a few examples of what can happen over here. So you can take a corrected value from positive to 0. So you can see that from 0. So initially it was 0, and then over here it gets corrected to something that is non-zero. You can also take something that was positive and then set it equal to 0, or you can take something that was positive and again get it to some sort of positive value. Again, all we're doing is we estimate the original quantile function, you estimate a corrected one, and then you match the corresponding quantiles over there. All right, nothing terribly complicated going on. All right, so operationally, how does this work? So recall, this is the CARDIA data. We have, after some processing, 380 taxa on 540 samples from 6 different batches. The key variable of interest in this case is systolic blood pressure, which is a continuous variable. And additional covariates that we're interested in looking at include age, gender, and in this case, I think it was dichotomous race. All right, variables. And so these are the principal coordinate analysis PCO plots before and after correction. So this is looking at Bray-Curtis dissimilarity. This is looking at Bray-Curtis dissimilarity of the original data. This is with using combat-seq, an approach developed for RNA-seq data. And then this one is after using Conquer. And so you can see that using combat-seq, an approach for RNA-seq data does seem to help, but it doesn't smush it together quite as well as Conquer. And the same is true on if we use a Euclidean distance instead of Bray-Curtis dissimilarity over here. Right? Everything sort of moves together more closely. It's not perfectly on top of each other, but it is close, much closer. all right? And you don't necessarily want it to be perfectly on top of there, all right? Now here, what we have are the Permanova R squared. So this is just a multivariate R squared with a variable before and after correction. And so here, this is the R squared with regard to the batch variable, and this is the R squared with regard to systolic blood pressure. So this is on the original data, all right? And so you can see about 10% of the variability in the data, at least using this particular measure of variability, comes from the batch effect. The effect of systolic blood pressure is like 0.3, 0.4%. So very, very modest relative to the batch effect. All right, if you use combat-seq, it does seem to help quite a bit. All right, and the effect of systolic blood pressure is pretty closely preserved. If you use Conquer, again, it knocks it down all the way to 0.05% variability due to the batch, and while still maintaining the same variability from systolic blood pressure. So the punchline of this slide here is that it does significantly reduce the batch effect while still preserving the signal of interest in this case. Right. Here's one where instead of R squared, we looked at prediction. So this is just the cross-validated prediction performance. And so you can see that it closely matches what we had before. And the RMSE after using Conquer is much better. There's not terribly too much interested in this, but every time I give this talk, all the microbiome researchers want to see a random forest. And so it's here. So that's with regard to dealing with a traditional batch effect, all right? The other type of analysis that one might be interested in this case is doing a mega-analysis where we combine data from different studies. And so this is from the HIV Reanalysis Consortium. So each of these studies, I think there are 10 different studies, we're studying the relationship between gut microbiota and HIV status over here. And so what we did was we aggregated the data from each of the different studies, which were each conducted with different sequencing protocols and designs. And then we treated each study as a separate batch. So this is much more challenging than traditional batch correction because of the different protocols and totally different designs of most of these studies. But we sort of ignored that and said, what happens if we just smush the data together, as actually the HIV Reanalysis Consortium had already been doing. So the pooled data included 606 taxa from 572 individuals from 10 different studies. And there's 10 different batches ranging in size from 24 to 135 patients. And so in this case, the key variable of interest was binary HIV infection status. And covariates included things like age, BMI, and gender. Gender was a very complicated variable because it was we were trying to use it as a surrogate for receptive anal intercourse, which is supposed to be one of the major drivers. But that was not collected in most of the studies, so it tended to be more MSX status going in. Right? And so these are the findings. These are the PCOA plots before and after correction. And so on the Bray-Curtis scale, you can see that there's huge differences between the studies overall. All right. If you use combat-seq, it does seem to help a little bit, but it does not seem to work as well as if you use Conquer. Now you'll notice in this case that the ellipsoids and the centers are not as good on top of each other as in the CARDIA data, and that's simply to be expected because there are huge differences and we're not able to fully correct for those. Right, and on the Euclidean scale, that becomes even more obvious that we're doing something, but it's imperfect. Nonetheless, if you look at the R squareds before and after correction, in the original data, 30% of the variability in the data was resulting from differences across the different batches, the different studies in this case. HIV status was like 0.5% of the variability, so very, very modest. If you do combat-seq, it does seem to help quite a bit. It drops it from 30% to 20%, but if you use Conquer, it drops it down to 1.2%. So that sounds really good, but bear in mind that the proportion of variability due to HIV status is still minuscule in this case and relatively small due to relatively modest as compared to batch. So Conquer helps, but it is not a panacea. It does not solve everything over here. But that's the extent to which we've been able to do anything here. So better than nothing, but still not perfect. And that's simply a limitation of our approach that we acknowledge. One of the reviewers asked us to find a study where batch effect was not as big a problem, and we just couldn't find one. Every single study that we looked up that had a batch effect, the batch effect tended to dominate every variable of interest that we were interested in studying. And at the end of the day, we found one that was sort of on the same order of magnitude. I think batch was like 10%, and the clinical variable was like 5%. But there's no situation that we found where batch effect is not the major driver when we combine across different batches over here. All right, so we can also do prediction, same story over here again, because people like random forest. So a couple of remarks on this part before I move on. Batch correction for microbiome data is increasingly important, especially as we get larger and larger sample sizes. There's simply no way to sequence all of the samples together at the same time on the same machine using the same reagents and the same technician. It's simply going to become impossible, especially if you start dealing with 25,000 samples at a time. Right? So Conquer is an approach that seems to work in many situations. It works directly on the count data. It does not make specific distributional assumptions, and it seems to remove the batch effects reasonably well, especially if you have a classical batch effect situation, while preserving the signal of interest. There's additional things which I didn't show, including regularized versions, which we've incorporated into here, versions that accommodate library size, as well as some approaches for tuning across different ranges of taxon prevalences. The limitations of Conquer, and actually of most batch correction methods, it doesn't work very well for super rare taxa. If you only have things that are super, super rare, all right, it doesn't hurt, but it also doesn't necessarily help that much. We tend not to worry about that quite as much because there's also no power in those situations, but nonetheless, that is a limitation of our approach. If your batch effects are really, really small, so suppose in the worst case, every sample is in its own batch, then there's absolutely nothing that we can do. So we do require that you have to have a certain number of samples in the batch. Bigger, of course, is better. It does also correct each taxon individually, which does not directly acknowledge the correlation among different taxa. And we are double dipping. We include the variable of interest in the model. And that is something to be concerned about. A reviewer raised that question, and they're absolutely correct. We're already using that variable, and later if you do association analysis, in principle, your standard errors are going to be too small. In practice, we have not observed that, and this is something that all batch correction approaches combat everything. All of them do it, and it doesn't seem to be a huge problem, but we acknowledge that there are situations where this could be a problem. We simply have not run into it. And in fact, our simulations and real data analyses seem to suggest that doing Conquer is way better than not doing anything. All right, with that, I'm going to move on to very quickly, because I'm running short on time, talk about the second part of this presentation, which is about how we robustly identify individual taxa associated with outcomes of interest over here. So recall now that you have huge sample sizes, and so now we're trying to associate the microbial taxa with exposures or variables of interest. So association analysis is a recurring objective in most microbiome studies. And so the usual approach for screening for individual taxa associated with variables of interest, you normalize or scale the data. So you make everything have norm 1, or you normalize to some other quantity. And then you assess the association between each feature, each taxon, and the variable of interest. You get a p-value, you would control for multiple testing somehow, and then you follow up with auxiliary analyses. Things like network analysis is almost always done, or other fun things that you might be interested in doing. All right. And so with regard to microbiome studies, there's basically 2 major classes of approach for assessing the association with individual taxa. The first one is to apply the methods that we all learned in kindergarten. All right, so that's to say you take your t-test or Wilcoxon test. All right, if you want to get a little bit fancy, you take something from first grade like linear regression. All right, but you basically are using very, very classical approaches. The other class of approach, all right, is to use what we call tailored approaches. Approaches which are tailored either to genomic data or to microbiome data specifically. And so examples of classical approaches there, all right, include, say, the t-test, Wilcoxon test, regression. All right, these are great, but they tend to lose a lot of statistical power over here. They lose a lot of power in this case. They also struggle with zeros and ties, and they're not taking full advantage of the characteristics of the data. That being said, they do usually protect type 1 error, but they are simply not, they're not very powerful in many cases, right? In contrast, all right, there are approaches that are tailored to the approach. Right? And these approaches oftentimes have very, very poor type 1 error control. So one of the most commonly used approaches is DE-seq2, which was developed for RNA-seq data, and it has absolutely terrible type 1 error control for microbiome studies. It assumes that the data are negative binomial, and in fact, microbiome data are not negative binomially distributed. And so your type 1 error rate tends to be through the roof. There's other approaches such as metagenome-seq that basically normalize the data and then assume a zero-inflated Gaussian model. And again, that tends to be pretty bad. And so this is something that other people have observed as well, and that really motivates the need for new approaches. All right, so this is just 3 out of the dozens of papers that have all shown very poor type 1 error control for most of these tailored approaches. And one of the major challenges is that they make these strong distributional assumptions. DE-seq2 and EdgeR assume negative binomials. Metagenome-seq assumes a zero-inflated long Gaussian or Gaussian model, which again doesn't work. And then there's other zero-inflated approaches, such as zero-inflated gammas, zero-inflated betas. People are using zero-inflated negative binomials, Poissons, beta binomials. None of them really captures the distributions very well. And even if it works for one taxon, it does not necessarily work for the next taxon that you're looking at. So most of these approaches tend to have terrible type 1 error control. Right? And so this motivated the development of Zinc, which is again just using the same 2-part model that we had before. So what we're going to do is we're going to do simultaneous conditional quantile inference for zero-inflated data. So what we're doing is we're comparing the distributions of the data between, say, cases and controls, or in relation to a variable of interest over here. Okay. So what it is very, very straightforward. You're summarizing the difference at 0 counts and also at a grid of quantiles for the non-zero component. And then we have a rank score-based test in order to actually do the testing in this case. Right? The main advantages here are it makes limited distributional assumptions, and it still allows for covariate adjustment. It's regression based, and it can allow for discovery of heterogeneous effects across quantiles, which I'll talk a little bit more about going forward. It's one of these things that people in quantile regression always talk about, but we rarely see. But in microbiome data, it really does occur. Right? So it's the same 2-part quantile regression model that conditional quantile regression model that we had before. So again, you use a logistic regression component for whether or not it's positive or not. And then for the other part, we have this conditional quantile model. And so the idea is that then we want to test whether gamma, which indexes the importance of the covariate of interest C, whether that guy is equal to 0, or whether the beta tau function over here is equal to 0, tau being the particular quantile of interest, while adjusting for these Z's, which are additional covariates. Okay. So to test the distributions, again, you're testing your gamma and your betas, whether they're all equal to 0 versus the general alternative over here. So it's a global test. So the testing strategy is very, very straightforward. To test the 0 component, all right, it's simply a logistic regression over here. And so you can test for it using any valid test, walled score, likelihood ratio, anything that comes out of your logistic regression. For the quantile-based test, we test across a grid of quantiles, but it actually has to account for bias from the fact that we're only using the positive subset. If you just directly compare quantiles and ignore the fact that there's zero inflation, then you end up with some bias over there. And then but assuming we can do that, then what we do is we simply combine the p-values from the logistic test and the individual quantile tests and get a single omnibus p-value for the association with the clinical variable of interest here. All right, so here's a schematic of what's going on. What the input here is just a single taxon abundance data. It can be normalized or not. All right, and so we use a 2-part model formulation, logistic for dealing with whether or not it's 0 or not, and from there you can test for whether the effect of the covariate is equal to 0, just using a standard test. For the quantile regression part, you fit the model, and then we do a series of quantile rank score tests that are adjusted to account for bias due to excluding the zeros in the quantile estimation over here. All right. So all of these, from this, you get a bunch of p-values, which we then combine using either a minimum p-value approach, or we can do a Cauchy combination test. All right, both of these guys seem to work out. They work better in different situations. So this, for the most part, is just your usual rank score test, but then we adjust for inflation at the top quantile. I'm going to keep moving on. Right. The combination, again, this is just a simple minimum p-value approach, or you can use a Cauchy combination of tests. So the minimum p-value has basically lets the smallest p-value index the overall significance. This is useful if there's a single quantile where things are very different, and a single really small p-value driving things. The Cauchy test is basically like averaging the p-values, and so this tends to do better if you have a whole bunch of things that are different, but modest, right? So we can apply this to the CARDIA data. And so the outcome in this case is the normalized. So here we consider either rarefaction or cumulative sum scaling, which is rarefaction means you subsample so that everybody has the same depth, sequencing depth. So the total read count is the same for every subject. And then CSS just means that we normalize to some median cumulative sum scaling value. So some measure of central tendency in this case. Covariates in this case that we included age, physical activity score, dietary score, and systolic blood pressure, and hypertension in this case, binary hypertension, which we were interested in assessing for association with taxon abundance. So first, here are just some type 1 error results over here. So here we just permuted the data, and I've got a ton more simulations. And you can see over here, so what we compared is corn-cob, which is the beta binomial, DEC2, and HR, which is a negative binomial. The linear decomposition model is a slightly different animal. Lemma and linear regression. Monocle, which is used for, I think, single cell data. Q-rank, which is a quantile-based approach that does not take into account zero inflation. And zinc, our proposed approach using Cauchy combination test, as well as zinc using a minimum p-value approach. All right, and so you can see that on rarefied data, all right, a lot of these approaches that make parametric assumptions tend to do very, very poorly overall. On cumulative sum scaling data, so this is now on a continuous scale versus a count-based scale. Again, we see that, say, metagenome sleep, which makes distribution assumptions, tends to do quite poorly overall. Right, so those parametric approaches that are tailored to this situation simply don't work very well. Power, again, this is so here we have type 1 error and power actually. So this is false positive versus true positives. Where we fit this from real data. Where we fit the data where we simulated based on real data. And so this is zinc over here. It tends to, and it has pretty good, reasonable power in this case. Some of the other approaches look like they're better, but then their type 1 error rate is also through the roof in many of these cases. Right. So we applied this to the actual CARDIA data, and these are the number of differentially abundant taxa by the approaches that correct that have valid type 1 error in some situations. And so overall, zinc using the Cauchy combination test tended to find the most, call the most as differentially abundant. Some of the other ones also seem pretty good, but just bear in mind that they may have inflated type 1 error in certain situations. Okay. This is simply a Venn diagram, and you can see that overall, the approaches that are identified by zinc, the blue circle, tend to be zinc tends to find all of the ones that are identified by others. It's not perfect. You've got a couple that are straggling out there, but it tends to find the same things as the other ones. This is on the rarefied data, and this is on cumulative sum scaling data. In terms of patterns of abundance profiles of some of the taxa exclusively identified by zinc, all right, one of the interesting things is that it can discover heterogeneous effects beyond simple mean effects. So this is an example looking at Eubacterium, and for this normalization, we simply rarefied. So we subsampled everybody at the same depth. So the solid black line is the quantile function for the hypertensive subjects. The red solid line is the quantile function for the non-hypertensive subjects. The dashed line is the mean for those that are hypertensive, and the dashed red line over here is the mean for the non-hypertensive subjects. And so this is a situation, you can see that the means are actually fairly similar over here. But as you go to the higher quantiles over here, you can see that there become much more substantial differences. So this is a situation where looking across the distribution, you tend to do a little bit better. And you can see that the zero inflation rates are fairly similar over here. Overall, however, you do see that there's a general shift in this case. On the other hand, here's another one, Haemophilus, in which we looked at cumulative sum scaling data, right? So same thing. And the reason why you only see a red dashed line and not a black one is because the red dashed line is covering the black dashed line. So the mean abundance is exactly the same between hypertensive and non-hypertensive subjects in this case. But this is a case where if you look at the lower quantiles, all right, the non-hypertensive patients, their distribution function is actually above that of the hypertensive subjects. But then there's this weird crossover effect over here. And so if all you're looking at is in the middle, you're never going to see this. But if you look at the entire distribution, all right, it is clear that there is an overall difference. And so this Haemophilus is actually a very, very interesting taxon in this case, in the sense that Haemophilus is a genus. Below that, there's a whole bunch of different Haemophilus species. Some of them are thought to be related to hypertension risk, versus others are actually thought to be protective. And so those types of heterogeneous effects, they sort of smush each other out. But on the other hand, when you start looking at the overall distributions, you can start teasing those apart, right? Let's skip this, I didn't like that slide, right? So overall, reliable testing of individual taxon effects continues to be a very challenging problem. This is something that we face day to day, and there are more papers coming out weekly on that first outline how terrible existing approaches are, and that in some cases propose new models, right? We didn't compare to all of them just because literally more are coming out every week. And it's really hard to keep up with that. Zinc is one approach that we think works reasonably well. All right, it's quantile-based differential abundance, differential distribution test. It's robust to distributional issues. It works under different normalizations because it's just comparing distributions overall. It tends to have better type 1 error control than many of the tailored approaches, but better power than many of the classical approaches, and can also capture heterogeneous effects in many cases. The limitations of this approach are first that if you have a lot of for low frequency taxa, it doesn't do that well. But this is again a limitation of virtually all methods. If you only have a couple samples in which it is positive, you're simply never going to have any power to do anything with that particular taxa. The other limitation is that we're assuming we're dealing with a situation where we have large sample size. If your sample size is very small, our approach tends to be conservative. And that's simply again one of the limitations. But as we move towards these large scale studies, this is a potential approach that can be used. One other final limitation is that we don't necessarily have a nice effect size of interest over here. So when you do linear regression, you get your betas, logistic regressions, you get your betas. We don't do that, we don't get that here. It's like a Wilcoxon test. It's just you get a p-value out of there that says this is the overall difference. You're not really getting a sense of specifically what is driving that. So again, those are simply the realistic limitations of our approach. But if you're willing to overlook that, we tend to think that this approach is quite useful. So just some acknowledgements. This was driven by a former super minion of mine, Huodan Ling, who's now on the faculty at Cornell. And there's tons of statistical collaborators as well as folks who are involved in generating the data. This is my funding for these projects. And here are some references. The Conquer approach just appeared last week, and Zinc appeared last year in Microbiome. And finally, a brief plug, I'm hiring for multiple postdoc positions. If you're interested in one position is about microbiome analysis, one is on genomics for cancer clinical trials, and one is a broad position looking at statistics and data science within the context of cancer clinical trials. We're looking for people who are interested in doing trials designs, as well as folks who are interested in just mining the 30+ years worth of clinical data that we have with our community. And the last position is not necessarily working with me, but could be with others. With that, I will stop here and take any questions for anybody who still remains awake. All the students here who I put in cages. All right, I really wouldn't do that in real life. Cages are expensive.
Very interesting talk. So I have two questions for the first part, the taxa fact question. So you are doing it for each taxa segment.
Yes.
So that is to say, for different taxa, essentially you can have different taxa fact correction.
Yes.
So do you worry about overcorrection in that sense?
We always worry about overcorrection in that sense. And that's why we have to stick in the variables that affect taxon abundance. So things like covariance of interest. You're right, we're basically smushing everything together. And so we do worry about overcorrection. But the overcorrection that we're worried about in this case is not that we're that we're smearing out signal, but that we're overfitting our own data. And so our standard errors are going to be too small when we then do the association analysis. But at least in our simulations, that hasn't really panned out. Our type 1 error rate, actually, I was kind of surprised, still is on point in this case. People have raised this concern with other batch correction procedures in the past. I think there was a biostatistics paper that raised this point with combat and some of those other approaches. But regardless of that, these approaches are still widely used. And I think most simulations that are more realistic rather and not exaggerated tend to show that these approaches seem to work okay despite that. But I agree, that is a concern in that it does affect the theoretical validity of our approach. But in terms of practical application, you got to do something. And if you have an alternative, I'm very happy to incorporate it. But in the absence of that, we think that this has some promise, at least at this point.
Sure. Yeah. And another related question is sort of an important thing that you need to decide is the quantile levels, right?
For the first part, no, because we just search across a very fine grid of quantiles to generate the entire quantile function. All right, and then once it's super fine, then a little bit of messing around doesn't affect it too much. You can interpolate a little bit if you really want to make it faster. But for the second part of where we're testing, yes, you need to pre-select the quantiles at which you test. And that's going to be a function of your sample size. If you only have 100 samples, then maybe you'll pick like say 3 quantiles to test compare across, versus if you have say 10,000, then you can actually compare at a relatively fine grid in that case. So maybe every 10 or so. But you're right, that does have to be pre-selected. And that's going to be a function of sparsity and not sample size, sparsity in this case, and sample size.
Thank you.
Maybe a follow-up from the first question.
Yes.
So it's clear your definition of batch effect doesn't include unobserved confounding.
Yes.
Right. So that being said, I'm curious about the strategy using batch labels to combine multiple studies. Isn't the meta-analysis the right way to deal with multiple studies in literature?
I think you're right, it probably is, but it's actually very interesting. In the microbiome literature, they call this type of mushing data together meta-analysis as well. I keep telling them it's mega, but they disagree with me. But anyway, that's a side of the story. But you're right, it probably would be better to, in some cases, to do that. But one of the advantages of this is that it allows you to visualize data together in some cases. So that's why I like a batch removal sometimes a little bit better than batch adjustment. It allows you to do other things that you might be interested in doing by pooling the data.
Right.
But if all you are doing is association analysis, then in those situations, yeah, you might be better off either somehow adjusting for it or analyzing each study separately and then merging the analyses that way.
Yes, I guess the point is do you think that the label itself is sufficient to account for the study specific unobserved confounding?
That's a really, really good question. Actually, I think a reviewer raised that question. I can't remember what my postdoc said, but that's a really good question. We do assume that if you have batches within each sub-study, then you would have to include that as an additional variable. And so you can break out batch further, but we require knowledge of the batch effects in this case. So in situations where batch effect is unknown, we are actually working on that problem. I think for gene expression data, methylation data, we use surrogate variable analysis, Jeff Leake's approach.
Right.
And that's a batch adjustment procedure. It's one that's useful for adjustment within the context of association analysis, but it still doesn't actually allow you to correct it out in any meaningful fashion.
Yeah, I agree with that assessment. But probably because the batch definition is a little bit different. It includes unobserved.
Yes, you're absolutely right. We're playing around with it, but it's actually really hard within this context to do it. And if you want to talk about that, I'm happy to discuss further. But that's a really good point that you're right. We do assume that we know all of the batch variables. If again, there's some batches within each study, we can incorporate that. But if you don't know it, then unfortunately, there's nothing I can do in this case. That's going to be another limitation.
Yeah, I have a similar question here. So when you combine this data from different studies, you use the same quantiles, but what I'm thinking that when you have the taxa data from different study population, maybe some younger and older, the quantiles at the same quantile level may mean something different.
Yeah.
When you combine them, either use mega or meta, does that make sense? The same fix the quantile because the distribution.
I mean, at least when we're trying to mush things together, we try and adjust for each quantile for age. So if you have, participant age, we can stick that in there.
Right.
How well it's estimated, I don't necessarily know, but we would at least stick it in there.
There must be an assumption behind it, so that same quantile position that somehow homogeneous across different studies.
Yes, you're also right that we're assuming that the effects of different variables is homogeneous within those populations. You're right. That is a separate assumption in this case. And we can adjust, but that's still not going to deal with the fact that if it's heterogeneous across there, we're not able to correct for that at this point. So that's a really good point, and that's probably another limitation that I should list here. Yeah. Unmodeled batch, that one, I don't know what to do with. This one, that's again, one that we'll have to think a little bit more carefully about. It may be possible to add in some additional modeling in that case, but if some of the batches get small, it becomes harder. So, good points. Al, excellent points. So all the way back.
I was just curious about the choice of the combination test. So is there one obvious choice, like under certain scenarios that one is preferred other than?
Yes. So the good question. So in general, the Cauchy combination test is sort of conceptually averaging the p-values. So in a situation where you think that there's a bunch of mediocrely small p-values, all right, then mushing them together using the Cauchy combination test as an omnibus approach tends to do well. If there's a single quantile where you think there's huge differences and everything else is basically the same, so you have one really, really small p-value, then looking at a minimum p-value approach will tend to do a little bit better. In general, simply because you're simply taking the most significant thing in this case where there's a huge effect versus averaging everything, which can again smear out your signal. But excellent question.
So for the batch correction on the BC distance you mentioned in the first part.
Yes.
Do you correct the BC distance after calculating or?
No, so what happens is we correct our raw data, the raw counts, and then we estimate the BC distance in this case, or dissimilarity. So everything is done up front. The idea is that we do everything up front, and then afterwards you can do whatever you want with your data. And that's one of the differences between what we're doing and the batch adjustment approaches. One of the things was visualization. How do you visualize the data better, given that you do have all of these batches that are smearing out the differences between hypertensive and non-hypertensive patients?
So I thought this data, microbiome data, was compositional. So they add to 100.
Yeah.
And so I'm understanding you're doing each one separately and then
Yes.
you're adjusting. So do they still add to 100 afterwards, or does that matter?
We don't start off with them adding up to 100, actually. We just start from the raw counts. So we don't normalize. So they only become compositional after you normalize. And so you force them to have norm 1.
You have to normalize, no?
We don't have to actually up front. I mean, the normalization is one of those things which, first of all, the compositionality I've got some issues with. I think that people care way too much about it, way more than they need to. If you have 1,000 taxa, then that sort of correlation really becomes pretty minuscule, especially since in the gut, you tend not to have dominance by any particular taxon. The On the other hand, if you're dealing with a vaginal microbiome where there are certain dominant species, then you have to be a little bit more careful. But most of the time in the gut doesn't matter. But ignoring that, we start from the raw count data. So there is differential read, so the read depth for each sample is different. And we simply, it does tend to be batch specific though, that overall within a batch, it tends to be fairly similar. And so that's why we sort of ignore that. We do have an approach where we adjust for the differential library size upfront and we model that in there. And the results have been mixed as to whether or not that really does improve things. It's basically becomes this this additional offset in sort of a Poisson type thing, and it's in the actual paper. I didn't talk too much about it because I don't like it very much, but it is something that reviewers ask for, and so you're right, we've included it, but I don't think it actually helps that much. But absolutely fair. We're not starting from the compositional data in this case. It's actually kind of interesting because we do batch correction and we're just sort of making the distributions look more similar. The actual effect of differential read depth becomes much more modest as well. So it's inadvertently also doing a little bit of correction for library size.
Can I ask another one?
Questions.
So I think what they're trying to achieve here is to present a clean, like batch correct data.
Yes.
But I mean, it's sort of like a imputation where you have some missing data that you try to impute once and you give some data for other people.
We do GWAS all the time, right?
So I wonder if you have multiple bias correct, like batch correct data, because you said the data double dipping or some like sort of overly small
Yes.
standard errors or something like that. If you can, you still have some variation in bias correction. Can you somehow simulate different types of this bias correction and generate multiple data set of like similar idea to multiple imputation? You don't do one data batch correct. You have multiple of them and try to generate a little bit the variation there to mitigate a little bit sort of single batch correction sort of decision making in the data cleaning. I don't know if that can help you to, because you said you do not observe much of the
Yeah, we don't
difference
observe
between
any actually.
But I wonder.
Theoretically, it is a problem.
Yeah.
No, you're absolutely right that theoretically it's a problem. And that's a really interesting idea. I guess it's conceptually possible. You assume a distribution on there and then you correct it across a distribution of different batches somehow.
That's like multiple imputation.
Yeah, exactly. And I'd have to think very carefully about it, but that's a really interesting idea. So I may pester you more about that, but I haven't thought
I'm just
carefully about it.
trying to find some solution for your questions.
Yeah, no, I don't have a good solution. And so if you do, this could be something very, very interesting. Yeah, but see, this is why I like giving presentations here. All the people are smarter than me. Not all of them are as scary as Jeremy, but I mean, it's yeah.
Do we have any question for online or?
Oh, there is an online.
Yeah, I think it's just for recording.
Oh, just recording.
So I'm happy to stop here. Make sure if you're an 8bio 800 that you sign the sign-in sheet. And don't sign in for your friend. Not allowed. All right.